Hello, everyone. This is going to be the second half of chapter 7, which is cell transport. So we're talking still about the cell in this section, and I like to divide this chapter up into two, just like I did the last chapter, just because there's so much information here, and this is a different kind of thought process. So in the last part of the chapter, we talked about organelle structure and function. In this chapter, or this part of the chapter, we're going to be looking at how things get into and out of the cell, and how that affects then the size of the cell, or just how the mechanics of the cell go on inside. So without further ado, right, passive transport is going to be the movement of materials into and out of the cell from areas of high concentration to low concentration. This is called moving with the concentration gradient, since we're going from high to low concentrations. Passive, when you think of the word passive, think of no energy needed. So we don't need energy for this to occur. This occurs on its own. So you can see here, I do an example in class where I put a drop of food coloring in a beaker, and after a while, without me stirring, without me adding energy, that drop of food coloring is going to color the whole beaker. What happens is that reaches equilibrium, where there's an equal distribution of the molecules. And again, this is because things just tend to move away from each other, and it's just the high concentration to low concentration movement that we're talking about with passive transport. Now, there are three different types of passive transport that we're going to talk about: diffusion, facilitated diffusion, and osmosis. Let's get into each a little bit more in depth. The diffusion, this is the movement of specifically molecules. You can see here we were going from high concentrations to low concentrations in these two animations. This one's going to end here at equilibrium. You can see here it's at equilibrium where they're evenly spaced out. You saw over here we had a high concentration of these red dots. They're going to move and equal out and reach equilibrium here around, right? Yeah, it's about equilibrium right about now. But that is the diffusion. We're looking at molecules specifically. Next is facilitated diffusion. The only change that with facilitated diffusion, again, does not require energy. We're just moving through membrane proteins, where we're going from areas of high concentration to areas of low concentration here. And the first one that we talked about, diffusion, that's just in any space. So it could be in a beaker, could be just in the room, whatever. So facilitated diffusion is through a membrane. That's the big difference. That's actually the only difference, is you're moving across this semi-permeable membrane. Let me go back. Osmosis here is the movement of water over a selectively permeable membrane. So now we're only looking at water. Water is again going to move from areas of high concentration to low concentration. As you can see in this beaker, we have this semi-permeable membrane in the middle. Here we have more water molecules than the red solute molecules that are sugar. You can see there's a higher concentration of water here compared with here. So water is going to move from the right to the left, and that's what happens. You can see after osmosis takes place, water levels on this right-hand side are going to drop, and water levels on this left-hand side are going to rise, because water is moving from the right side to the left side. This is called osmosis. Let me give you another example here. We have a much higher concentration of water on this side than this side. So what's going to happen is we have this selective permeable membrane. Water is going to move from the left to the right. So after a while, you're going to see the water rise on this end and the water lower on this end. So again, this is osmosis, and this is only through a semi-permeable membrane. This is just the movement of water. Next, with osmosis, we're going to talk about isotonic, hypotonic, and hypertonic solutions. So isotonic, this is when there's an even amount of water and solute in the cell and in the water that surrounds the cell. That's called an isotonic solution. Hypotonic solutions, I think of the big O for hypo. What's going to happen is there's a higher concentration of water outside the cell than inside the cell. Water is going to rush into the cell, and it's going to make the cell swell. So that's why I think of the O for hypotonic. Hypertonic solutions are like really a solution with a lot of like solute. What's going to happen is there's a higher concentration of water inside the cell than outside the cell. So water is going to rush out of the cell, and it's actually going to make the cell shrink. So you can see here in this next, right here. So we have water going into that hypotonic. So think of that big O for hypotonic. Isotonic solutions, there's going to be an even exchange here. You can see the even amounts of water going in and out because the solute and water concentrations are the same. And then hypertonic solutions, think smaller hyper. You're having water leave the cell because, again, there's a higher concentration of water inside the cell than outside the cell. Here's what it looks like in real life. You can see the hypertonic solution here. Water's leaving. Isotonic, there's an even amount. And then hypotonic, there's more water going into than out of the cell. All right, so that's done with passive transport. Moving on to active transport. This is when you're usually moving, molecules across a plasma membrane. This requires the input of energy, and sometimes this is from areas of low concentration to a high concentration. But the main thing here is we're using energy to move materials around the cell. Three kinds of active transport we're going to talk about in here: active transport pumps, endocytosis, and exocytosis. Endo and exocytosis are pretty easy because they sound like what they do. Active transport pumps are a little bit more difficult, but I really don't have you kind of go in depth with these. This is, again, we're looking at how. Let me go back here. We're looking at how membranes are going to kind of pump molecules across the or against the concentration gradient. You can see we have this low concentration here at the bottom and this high concentration here on top. It's going to move from areas of low to high concentration, and it needs ATP or energy to do that. Endocytosis, think endo meaning into the cell. So endo, this is when a large molecule or like a group of molecules is going to be folded. The membrane is going to actually fold around the molecules or whatever is trying to get into the cell, and it's going to form, if you remember that word, vesicle. It's going to form a vesicle around that substance. This is one way that molecules get things into the cells. They just wrap their membrane around it and just intake it. Excuse me. And exocytosis, let me go back. Exocytosis is the exact opposite. Think exo with exiting. The membrane of the vesicle is going to fuse with the outer membrane, and it's literally going to push that contents out and push, this is how the material in the cell gets out of the cell. This is how waste products get out of the cell. This is how a cell just pushes things out that it doesn't want. So remember, this is endocytosis, where the molecules are coming into the cell and getting wrapped around with the vesicle or the membrane. And this is how exocytosis, the molecules then leave the cell. This is all for cell transport. There's not a lot of information here. I usually go this in over 1 and 2 days. So if it seems short to you, yep, it's not a long portion of the chapter.